Okay, so this is chapter 15, and we're going to talk about the innate and adaptive immunity. So, what we're really going to do is kind of go over the immune system. We're going to briefly go through the innate immune system because you've already learned most of it, or you should have. So, to start with, this is actually from chapter 14. We're not directly covering chapter 14, we're not doing any prep you or anything for that, but these are things you probably should have learned in microbiology, but I want to go over them anyway, just so you're familiar with the terms. Prions, so these are types of pathogens. Prions are basically proteins that fold other proteins. That's the way you can really think about that. But you have in you right now, you have proteins that help fold other proteins. Prions are these kinds of proteins that have gone bad. And so they're helping fold other proteins, but they're really not helping. They're folding them wrong. And so the way that's defined is that they're infectious host proteins that can alter other functional proteins. So this is not that unusual that you have a protein that helps fold another protein. In this case, they're just doing it wrong, and they can cause Creutzfeldt-Jakob disease, which is a brain abnormality. The mad cow disease, that's another one. Viruses, hopefully we understand viruses, a protein coat surrounding a nucleic acid core. They have to rely on the host. They don't have any metabolic enzymes of their own, so they have to use their host, which means they can't survive for very long with outside a host. Bacteria, I'm sure you covered that a lot in micro. Cells without membrane-bound organelles or prokaryotes can live independently. So these little guys can live on the surface of things and can come back and infect things later on. So these are on just about everything. They say that if you remove everything from the earth except the bacteria, you would still see the earth. So that kind of gives you an idea of how many bacteria there are and where they are. Release exotoxins or may release endotoxins. Exotoxins are things that they produce and then release into the environment around them. Endotoxins are things that they are made of that when they break down, they can cause an inflammatory response or something like that. Mycoplasmas, these are just small bacteria that they tend to be resistant because they lack a cell wall. And fungi, which usually fungi don't cause any problems unless we're immunocompromised, and then they can cause a problem. So most of them require cooler temperature than the human body core, and most infections are on the surface of the body. Parasites, these are things that eosinophils would go after. Protozoa, malaria. Well, malaria is that attacks red blood cells. We kind of talked about that. Amoebic dysentery, giardias, and these are the ones that eosinophils would go after: the helminths, the roundworms, the tapeworms, the flukes. Arthropods, ticks, mosquitoes, mites, lice, fleas. Okay, so we know what these are, and these are a little bit larger, and those can actually be vectors for other diseases. Okay, so how do they spread? Most of these just make sense, and we already know this. Direct contact with the pathogen. So somehow, like this guy sneezing, the pathogen can actually move in. We can inhale it, something like that, or touch it in an open wound, something. Inhalation, ingestion. So I guess this would also include the sneezing. So we can inhale them directly into our lungs. Ingestion goes straight into our GI tract. Contact with a zoonosis, that just means that animals that can spread them to humans. So most animal pathogens can't be spread to humans, but they're out there. Contact with a nosocomial infection, that just means hospital acquired. Okay, so we can see things like MRSA that can be spread a lot in hospitals. Contact with a fomite, which is an inanimate object capable of harboring pathogens. So that would include something like a stethoscope, but that's pretty much an endless list there. So the stages of infection: the incubation stage, you're not going to really know what's going on. You may have been infected, you may not have. Oh, you probably gave me your cold. Activation, active replication without symptoms. So you don't know anything yet. And then after a few days, I feel like I might be coming down with something. And that's where you get early signs and symptoms, like maybe a fever, maybe some fatigue. And then you know you have it, the acute stage. So at the acute stage, that's maximal manifestation, tissue damage, inflammation, whatever it is that's causing the infection or whatever the infection is, you're going to see the maximal manifestation, signs, and symptoms. Convalescent stage means you're getting it contained. So you contain the infection, you eliminate the pathogen, and you begin to repair the damage. And then the resolution stage is the last one, total elimination, no residual manifestations. Your immune system has eliminated it at that point. Terms for infection. So we've all heard itis. Itis just means inflammation, and it may or may not be due to infection. So emia means in the blood, so sepsis or septicemia means bacterial toxins in the blood. So we'll see that term from time to time. Virulence factors. A virulence factor makes an infection more virulent. So it makes an infection more likely to cause a disease. Toxins like exotoxins or endotoxins from a bacteria. So maybe the bacteria is using some of your resources. Maybe it's maybe it's even destroying some cells. But what makes it more likely to cause a disease is the fact that it's releasing these exotoxins or these endotoxins when it's being destroyed, and maybe that's what's causing more of the signs and symptoms and interfering with things more than anything else. Adhesion factors help an infective organism stick to the body. So some of these organisms wouldn't even be able to infect you except that they have developed ways. They've developed adhesion factors that will let them stick to you, whether that's the mucous membranes in the GI system, whatever, in the lungs, but they allow the organism to actually get to you and infect you. And so we would call that a virulence factor. Evasive factors to help keep the immune system from killing the infective agent. So, certain things like tuberculosis can live inside macrophages. So they have developed evasive factors to allow them to infect in a way that would kill most other pathogens. So all of this, you have to have something. The pathogen has to have something that allows it to infect and cause the disease. Okay. Microflora, so not all microbes are not pathogens. So microflora is what we refer to when we're talking about our gut bacteria or the microorganisms that live in the gut. And it's not necessarily the gut. Somebody asked me that, and it's really anywhere where there is access to the outside. And that includes the gut, because the inside of your gut is really the outside of your body. It hasn't entered yet. If you're like a donut, if you put a marble in your mouth, it'll go through the other end and it'll never enter your body. So there are a lot of bacteria there. The vaginal cavity, the anal cavity, the lungs, you'll have organisms that are kind of living in those areas. Most of them, though, are, yes, in the GI tract, though. Okay, um, so when we talk about a pathogen, we mean something that causes a disease. That's, by definition, that's what it is. A pathogen is something that causes a disease or throws us out of our homeostasis. It's somehow messing with things. But sometimes a pathogen would not normally affect us. So we sometimes give it the opportunity. And if our immune system's down, if we're sick with something else, sometimes an opportunistic pathogen will come in. And opportunistic pathogens are microflora that are capable of causing disease really only if your health and immunity are weakened. Okay? And so they'll become a pathogen if they're given an opportunity, but they normally aren't. And see, and this is why one reason why antibiotics can be a problem, because if you take a bunch of antibiotics, that may allow these opportunistic pathogens to get a foothold, and then they can cause a disease. Okay, so how are we going to keep these things out of us? The first line of defense is always barriers. Okay, so we just try to prevent them from ever getting in. So the skin is a big one. Okay, skin, it's a physical barrier. It's slightly acidic. It has all of this, it has lactic acid that kind of moves out in sweat and things like that, and that keeps the pH a little bit lower. So it keeps the pH at about a 5.5, which is obviously lower than the inside of the body, or most areas inside the body. And so it tends to destroy any bacteria that would be able to normally live inside the body. We also have friendly bacteria that live there. Okay, and those bacteria will block any kind of pathogenic bacteria. It's already sort of an occupied space, and so the pathogenic bacteria are unable to move in for that reason. We also have stomach acid, so if you eat the Oreo off the floor, you pick it up off the floor, as long as it's not in a, like a daycare or worse, a hospital, you wouldn't want to pick it up there. But the stomach acid a lot of times will kill most of the bacteria, not all of them. Some bacteria can make it through. But you're sort of gambling at that point that there isn't going to be a pathogenic bacteria and that the stomach acid will kill it if it can. Sometimes you just need the Oreo, so it's worth it. So mucus and cilia, this is something you see particularly in the respiratory tract, but it's a way for any kind of mucous membrane. So any kind of mucous membrane actually is producing mucus. Mucus has, it has salts in it, but it also has antimicrobial agents that are made that tend to kill anything. Because usually where there's a mucous membrane, it's because there's an opening, and anywhere there's an opening, that's an opportunity for a pathogen to infect. Okay, the cilia just kind of keep it moving out of the airways so it doesn't collect in the airways. But the mucus being produced is really what, in many ways, protects you from any pathogens getting in. Earwax is another secreted substance that can kill and kind of slow down any pathogens. Tears are constantly running across our eyes and keeping any dirt and microbes flushed away. But however, we know obviously that pathogens can still get through. Injuries can allow pathogens to have direct access. Pretty much anytime you break the skin with an object, you're going to introduce some kind of bacteria. And so so your innate immune system is going to have to deal with that. And we've already talked a little bit about inflammation and what happens in that response. Some bacteria can make it through the stomach acid. So we do have like probiotics and things like that. If you have enough of them, most of them may be killed, but some of them may make it through, and those will then take up residence in mostly the large intestine, but all the way through the GI system. And we mentioned this earlier, pathogens may compete with natural gut microflora to invade, and I mentioned this is why needless antibiotics can be a problem. We talk a lot about antibiotic resistance, and that's a real problem. But also, if a physician prescribes antibiotics and the person doesn't need antibiotics, they just have a virus or something like that, in many ways, you're not just appeasing the patient, but you may actually be causing some problems. You may actually be causing more harm than good, because if they do have any of these opportunistic pathogens in there, then all of a sudden you're killing off your microflora, the healthy stuff, or the stuff that your body knows what to do with. Some of it's helpful, some of it just makes no difference at all. But you kill that off, and then you suddenly give a pathogen a place to live, divide, and infect. Okay, so overall, we know this has to be true, otherwise nobody would get sick. But pathogenic bacteria have evolved ways to take advantage of weaknesses or to overcome these barriers. Okay, so what we want to talk about is the innate immune system versus adaptive immune system. So the innate immune system is it's non-specific. So that's one good way to think about it. It's not specific. It doesn't care. All it says is, you know what? You're not from this body. You're non-self. And so it will attack anything that is non-self. However, I have here, we have to clean up after pathogens, but we also have to clean up dead cells. So cells are dying all the time, and we don't really know where they're going to die, but we have to get rid of them. So that's also a responsibility of the innate immune system. These white blood cells, these macrophages, are kind of going around just chewing up anything that's in the way that isn't supposed to be there. So dead cells, foreign debris, anything that enters our body that's not actively growing and actively contributing, the innate immune system should take care of. Okay, so I mentioned innate. It'll attack anything non-self, as well as its own dead and defunct tissues. It is faster, and it's faster because it doesn't really have a big, long decision-making process. It says, are you self? Are you from this body? Or are you from a human, really? And if the answer is no, it takes it out. Okay, so they comprise any white blood cell except the T helper cells, so the T cells, T helper cells, the CD4 and the CD8, and the B cells. So really aren't anything that aren't these B cells or T cells are going to be considered part of the innate immune system. Okay, so that makes it fairly simple to distinguish between the two. Of course, the hard part is remembering how they function. The innate immune system does not distinguish between microbes, so you could have a microbe that was living in your gut perfectly fine, not harmful, nothing wrong with it, but it somehow makes it into your body. Your macrophages will take it out. Okay, and it would do the same with a pathogenic virus. So something that could cause illness, it'll take that out. Something that can't, is perfectly harmless, it'll take that out too. So it doesn't care. And the innate immune system is also responsible for the inflammatory response. So we talked about neutrophils moving in, releasing cytokines, this inflammatory response, we talked about that earlier, and that's part of the innate immune system. So the adaptive, we'll probably spend a little more time talking about adaptive just because we haven't discussed this very much at all. It's also called acquired. Okay, so some books call it acquired, some books call it adaptive. I believe your book calls it adaptive. So so that works fine. Adaptive immune system, it is specific. So it is specific. That means that it will see this bacteria as a pathogen and kill it. This one, it will not worry about. Okay, so to within reason. Okay, so if that invades a cell or something, then it's going to be seen as a pathogen. But they tend to be specific to a particular pathogen. It will eventually invade this one, but we'll talk about how it does develop that specificity. Now, this is something you really need to remember, and because it's in every textbook, and it's a very generalized type of term. But humoral can kill free-floating pathogens, and those are B cells. Okay, so we're going to talk a lot more about this, but this is a general category. Humoral, free-floating, B cells take care of those. Cell-mediated, so the idea is something hides inside the cell. So you can imagine somebody sneezes on you, the virus gets in, and then the virus kind of moves into your tissue, and then it says, oh, this is scary, and jumps into a cell and hides in the cell. Is it safe? No, it's not safe, because T cells, specifically cytotoxic T cells, will eventually find it. Okay, so free floating B cells. And when I say B cells, I really mean plasma cells, because B cells develop into plasma cells and release antibodies. T cells or cytotoxic T cells will kill pathogens that are hiding in cells. Okay, so here's kind of a picture. This'll actually make a little more sense, but epithelial barriers, we already talked about those. Phagocytosis, natural killer cells, we'll discuss those a little bit more as well. But these are all innate, and the biggest thing to remember is that natural killer cells, even though they're the lymphoid cell line, they're part of the innate immunity. The membrane or the complement and the membrane attack complex, we'll talk about that as well. And then the adaptive immunity, and we see here B cells and we see T cells, okay? Specifically cytotoxic T cells and B cells that develop into plasma cells, okay? So we'll talk about how those things work. All right, so key cells of the immune system. We've seen this graph, or not this graph in particular, but we've seen graphs like this. And so we've already kind of covered a lot of these white blood cells. Specifically, we've talked a lot about neutrophils, first responders, pus. That's neutrophils. Macrophages move in a little later, but they're big eaters. That's what macrophage means. And so they phagocytose and get rid of pathogens, foreign material. The one we haven't really talked about are dendritic cells. We haven't really talked about, we haven't brought up dendritic cells in this class at all. And dendritic cells are included in this particular table. That's why I put this here. So you can kind of start thinking, okay, dendritic cells are important for the innate immune system, but they also communicate with the adaptive immune system. So, so well, I have a whole slide on these, but they're not called dendritic cells because they're dendrites or anything related to neurons. They just have these projections that make them look like dendrites. Now, we also haven't really talked about natural killer cells, so I have a slide on that, so we'll talk about those as well. And remember, that was innate, so all of these things are the innate immune system. The adaptive, again, we're back to B cells and T cells. However, the macrophages, and we'll see how this takes place, macrophages and dendritic cells, even though they're part of the innate immune system, okay, so we saw them up here, they will kill a pathogen and then they will take that pathogen, or at least parts of it, and show it to T cells. Okay, so they're largely responsible for communicating what they're doing. So they're like, they're like the security guard or something that sees something going on and then reports it to the police station. So these guys will communicate with the adaptive immune system to launch an adaptive or an all-out systemic response. Okay, so even though they're not lymphocytes, they work with the lymphocytes for the adaptive response. Okay, so we can go through these fairly quickly because we've already talked about a bunch of them. Granulocytes, remember the granulocytes, they have these little granules. You can see them pretty well in the basophil and the mast cells here. So they have these little granules that release stuff like histamine, heparin, they release some cytokines that communicate, they release certain toxins that will kill whatever it is they're affecting, particularly eosinophils do that. So these are granulocytes. The main granulocyte that we always talk about are neutrophils. So they're also, you'll see this sometimes, they're also referred to PMNs or polymorphonuclear neutrophils. I'll refer to them as neutrophils, but you will see this sometimes on papers and maybe labs or something like that, PMNs. So you need to know that those are neutrophils. They predominate early in the inflammatory response. We already know this. We've already gone over this. They're the first responders. They ingest bacteria, dead cells, and cellular debris. So they're non-discriminatory about what they ingest or phagocytose. They're short-lived and become a component of the purulent exudate. So in other words, they become pus. All right. Other phagocytes, so other things that are eating things, phagocytosis, monocytes that turn into macrophages. We've also talked about these. Monocytes are produced, well, they're all produced in the bone marrow, but monocytes are produced in the bone marrow and then they circulate in the blood. Okay, so these will then migrate to the inflammatory response or the inflammatory site. So this is kind of a review, and then turn into macrophages. Okay, so we know this. Macrophages are they're found in almost all tissues. So you have macrophages scattered around through almost all of your tissues right now. And they're in there, they're cleaning up any kind of debris, anything that's just kind of left there. But upon inflammation, they will be recruited and some macrophages will typically arrive at the site of inflammation about 24 hours or later. Okay, so the neutrophils, yes, first responders, but then about a day or so, the macrophages start showing up and they start doing a bunch of stuff. Okay, well, the bunch of stuff is mainly eating them, eating the pathogen, tearing it into little bits, expressing it on the outside of its cell, and then going up to the lymph nodes and telling the lymphocytes what's going on. Okay, so that was a bit simplified version of their function. Phagocytosis, yes. Inducing of inflammation, yes. They send out signals and tell other macrophages that there's something going on, or other cells that there's something going on. They have signal proteins that activate. Okay, so induction of inflammation is a little more specific, but they have signal molecules that activate, signal proteins that activate and activate other immune cells. So kind of works together with the whole inflammatory response that we've talked about. General scavengers, clear out dead cells, dead debris, whatever. And this is what I was kind of mentioning earlier. This is where they tell the lymphocytes what's going on. So they're antigen presenting cells. So whatever they eat, whatever they ingest, they will then present that on their a major histocompatibility complex protein that we'll talk about, and then they show that to the T cells. Okay, and then this one that we haven't really covered. Okay, so it's still myeloid, but these are, well, wait, phagocytes. Um, yes, phagocytes are dendritic cells. Sorry, I was getting them mixed up with natural killer cells. Yes, dendritic cells are myeloid. They're part of the myeloid line. But dendritic cells get their name, as I said, not because they look like they have dendrites. They're not neuronal tissue. Don't get that mixed up. Don't forget that. Okay, these are white blood cells. Okay, so they're definitely white blood cells. In fact, they're myeloid white blood cells. We just haven't discussed them yet when we've been talking about myeloid blood cells. Um, they're usually found under epithelial tissue, so skin, and in organs. They capture foreign agents and transport them to regional lymph nodes, which means that they are antigen presenting cells. Okay, so similar to what's going on with the macrophages. They mature in the lymph node, take on antigen presenting roles. Have I said that enough for not having explained it? Antigen presenting roles, release molecules that help direct the adaptive immune system. Antigen presentation. Okay, and they also will release molecules that tell B cells or T cells to start dividing because something is going on. Okay, so I didn't mean to oversimplify that there. Okay, how do cells of the innate immune system recognize invaders without being selective? That's a great question. How do you know that this wasn't in the body? How do you know that this stray thing moving around isn't just one of our cells that decided to travel a little bit? And we are hardwired, and I say that and it sounds a little mysterious, but that means we have receptors that recognize certain things that simply are not human, that they're always associated with a bacteria or something like that, or a virus. Some of these things are lipopolysaccharides. We've all heard of lipopolysaccharides. I know you have if you took micro. Lipopolysaccharides, peptidoglycans, and then there are certain lipoproteins that are specific to bacteria. We've heard of all these, and we've heard of all these things because they only exist in bacteria. Okay? So if they only exist in bacteria, it's pretty safe. Specifically, if they don't exist in humans, it's pretty safe that if you see these things, there's a bacteria associated with it. So it's okay to take it out. Okay, we don't need to preserve the bacteria that's just kind of just nosing around in our tissues. We can, it's fine to get rid of those. Other things like flagella. Okay, so we have one cell in humans, and only half of the humans have it, and that's sperm, but that's it. That's the only place you're going to find flagella, specifically the molecule or the protein flagellin. So if you see flagellin and you're not in an area where sperm is being made, then it's pretty safe to say that that's probably an invader. Okay, foreign DNA or double-stranded RNA may be associated with viruses. Take it out. Okay, that's not something humans make. Now, so we have these things. They're called pathogen-associated molecular patterns, or PAMPs. Okay, so I know I remember it like Pampers. I don't really have any relationship. I just remember the word. It's a weird word, PAMPs. But that's what we're talking about. These lipopolysaccharides, the flagella, the double-stranded RNA, peptidoglycans. These are all PAMPs. Pathogen-associated molecular patterns. Maybe they're not pathogens. Maybe they are. Either way, they're going. They're going away. Now, how do we do this? Our immune cells have things that are called toll-like receptors. They're named after a toll receptor, was something that was found in a fruit fly, And so these are similar to those receptors, and they serve the same function, so we call them toll-like receptors. So TLRs, whatever, wherever the name came from, that's the name they are, that we're stuck with. So TLRs are the things that recognize PAMPs, and these are made by you. They're receptors that are in our DNA, and so we're ready. We're ready from birth, pretty much, to start recognizing these foreign molecules or these foreign pathogens. Okay. All right. So that's pretty much it for the immune system. Now or the innate immune system. I wish it was enough, all of the immune system, but that's enough for the innate immune system. This is the adaptive immunity. So we're going to spend a little more time talking about adaptive immunity because we haven't talked about it much. And like I said, attacks specific microbes, and we call these what it actually attacks. We call these antigens, or what it recognizes. We call those antigens. And it develops after exposure to a specific antigen. Okay, so if you have a bacteria and it has a certain protein on the surface, here I'll make these proteins that are out here blue. These can act as antigens. So that's something that the immune system might recognize. You might have a T cell over here, and there might be a T cell receptor that might recognize one of these antigens. Okay. And we'll talk about the system and how that works, but that's essentially what it's doing. It's finding specific antigens and saying, okay, I know who you are. I have seen you before, and so it will launch a defense against this specific pathogen. So it has to, obviously, if it's going to be specific, the specificity or the cloning of it has to develop after. The all-out defense of it has to happen after exposure. So it has to be exposed one time, and then maybe it can, once it recognizes, says, oh, there's something that's strange, then it can divide more and more. Now, I oversimplified this because really, remember those macrophages. Those macrophages are initially going to tell on it. Okay, so the macrophages are initially going to tell the T cells what's going on. Okay, so once we have this adaptive immunity, once we've recognized a specific pathogen, that's when we have this humoral immunity. And I know I'm repeating myself, but humoral immunity means it's floating around, and those are the antibodies from B cells. So B cells are associated with humoral immunity. I said it again. Cell-mediated immunity, T cells. T cells are associated with that. And then once it's recognized a pathogen, let's say you get a cold, and little by little your adaptive immunity will figure out this virus. It'll figure out specifics about this virus, and then it will launch an all-out attack against it. But then what it does is it produces some memory cells. And so the next time the same virus, now these viruses have a tendency to mutate, but say the same virus comes in, you're ready to go, and you'll take it out very, very quickly, which means that the response is faster and more intense upon repeated exposure. Okay, and we call those memory cells, or the things that survive and stay with us. Okay, so we've been talking about specificity, but how do we end up with specificity? I mean, if I look at one bacteria and I look at another, I might see the difference in shape or something like that, but I wouldn't be able to tell one bacteria from another. So your immune system has to. And on that bacterial, if you want to call it a cell, but on the bacteria, it has a number of proteins that it's expressing. Okay, and let's say that this is one of those proteins. Okay, this whole green area. So I'm going to erase this so it doesn't get confusing. But let's say this whole entire green area right here is the protein. Okay, so it's a protein, so it means it has a three-dimensional shape. And so we can call this whole thing an antigen. But the immune system doesn't just say, oh, a giant protein, I recognize the shape of that protein. It's more interested in details. like maybe a specific sequence of amino acids on that protein. And we call those epitopes. So, so these lymphocytes, these B cells and T cells, they make receptors that are responsive to certain antigens, but it's responsive to a specific epitope on that antigen. I know that sounds a little confusing maybe, but it's not too bad if you remember, that here's a protein, and let's say on that protein, because proteins have that three-dimensional shape, let's say this is one place that the immune system recognizes it. This is another place it might. This is another place it might, and this is another. Okay, or maybe this little tail here. These red areas that I just drew are all the epitopes. The entire thing is the antigen. Okay, so the epitope is a specific part of the antigen that the immune system recognizes. The presence of a foreign antigen, okay, so let's say we don't make this protein. So the presence of a foreign antigen activates lymphocytes to combat an infection. So if we have a T cell that recognizes, okay, so it has a little receptor, we have a T cell that recognizes that particular epitope of that antigen, well then that's pretty exciting for that T cell because what he's going to be able to do is replicate, okay, and make more of them because we already know one of these is in here. So now we have a whole bunch of these that can recognize this particular epitope, this particular antigen, and maybe you can eliminate it that way. Okay, so the presence of foreign antigen activates lymphocytes to combat an infection. That's the same with B cells, by the way, B cells and T cells. Okay, recognize these antigens. We haven't gotten to who's carrying the antigens, and they're being carried on MHC1, but we haven't talked about that yet. So active immunity occurs when the lymphocytes are exposed to foreign antigens in the body. Sometimes we have passive immunity, like when we receive antibodies made elsewhere, like for a particular treatment or something like that. A lot of times they'll uh, you can inject antibodies directly into somebody's body, or directly into somebody's blood to kill something that they aren't able to kill. Also, probably a better example, I don't know if it's better, but the placenta, antibodies crossing the placenta to protect the fetus and the baby after it is born, and mother's milk. Mother's milk provides antibodies. Okay, so I always like to use this analogy for antigen versus epitope. If we were at war with France, I don't know why we would be, but it works for this analogy. So France would be the pathogen, which would be like a bacteria. Paris would be like the protein that the bacteria is harboring. So that's the antigen. So that's like the protein that's in the bacteria. And then the Eiffel Tower would be an epitope on that particular protein. Okay, so we're at war with France. How do we recognize France? Well, Paris is in France. Well, how do we recognize Paris? By the Eiffel Tower. Okay, so I guess, in Las Vegas, I guess there's a replica of the Eiffel Tower, so that might be like an autoimmune disease or something where the immune system gets confused and attacks something that it shouldn't. And that's actually not a bad analogy either, because that's sometimes what happens. Okay, so T cells recognized compromised cells, but in a very specific way. So this is where we get to talk about major histocompatibility complex. It's also called human leukocyte antigen. If so, this responds to a particular epitope of a specific pathogen. Okay, so this is the recognition that the T cells do. Okay, so they have these little receptors on the outside. They're called, handy enough, T cell receptors. Um, but the thing is, they're not just they're not just looking for pathogens. They're looking. Okay, so if we have tolerance to self-antigens must be provided by this thing called major histocompatibility complex. All cells in your body, so let's just say this is just a regular cell. It could be, it's round, so I wouldn't call it epithelial, but it could be a liver cell or something like that. All cells in your body have something called major histocompatibility complex 1. Okay? And what that does is it holds on to antigens that are produced inside the cell. So the cell is constantly sampling its internal contents. Okay, I have a better picture of that. But it puts those on this MHC out there, so we could make that maybe a different color. Puts that on the MHC, and then maybe or maybe not a T cell receptor will find it. These MHC, major histocompatibility complex, are really, when we talk about antigen presentation, they're the proteins that are holding on to the antigen, and they're different for everyone. Human leukocyte antigen. So that means that immune systems of someone else would recognize this and potentially wipe it out, okay? Because it's on every single cell. Now, if you're getting an organ donation or something like that, you want to make sure that this protein that is expressed on every single cell of the body, you want to make sure that it is as close to, let's see, if you're getting an organ transplant from someone else, you want to make sure that the donor has as close to a match of this as possible. And so that's where it gets its name, histocompatibility, tissue compatibility, or human leukocyte antigen. Uh, Major histocompatibility complex proteins are a primary marker for organ donation compatibility, as well as being involved in autoimmune diseases, which we'll talk about that later. Gene recombination occurs to create T cell and B cell receptors and antibodies that do not attack self. So that means that this T cell, if it's looking at something right here and it says, okay, is that self or is that not self? Now, it was produced by cleaving things inside the cell itself, but it's saying, well, now where did that come from? Did that come from a pathogen, or was it just a regular protein? If it was just a regular protein that the body already makes, it ignores it and moves on. If it's something from from outside, non-self, then this receptor and these receptors change, and they change form. Now, one cell will produce the same receptor, but it's very specific, and it definitely won't recognize that if it's self. And so if it does recognize it, which, boy, this looks really incriminating, all those arrows pointing at it, looks like it does recognize it. But if it recognizes this antigen, then it will destroy the cell, or it will be activated. Okay, one or the other. Depends on whether it's a helper T cell or a CD4 or a CD8. So I just gave an example of a major histocompatibility complex protein and how it can associate with a T cell. Now, all nucleated cells have MHC1, so I mentioned that. MHC1 will only be recognized by CD8. Okay? CD8 cells are cytotoxic. So if we go over this little exercise again, okay, and we see that here's a cell, any nucleated cell, and it expresses on its MHC1 a pathogen. Okay? Whatever that pathogen may be. Okay? A cytotoxic T cell will recognize that that's MHC1. Cytotoxic T cells are CD8. CD8 cells. So CD8 recognizes MHC1. And then it will sample and see if that's a pathogen. Okay? And if it's a pathogen, this CD8 cell, they're called cytotoxic. That's a pretty incriminating name. It will kill the cell. That's what cytotoxic means. So if it recognizes this as being foreign, that means that inside here there's a pathogen hiding, and we have this process where we kind of take proteins apart at random. You know, it's not selecting this particular pathogen and expresses it on that MHC1. And then the CD8 cytotoxic T cell recognizes it and says, oh, if I recognize you, you're non-self, and will kill that cell. Okay, so CD8 cytotoxic T cells recognize all nucleated cells and will destroy them if they present something that is non-self. Okay? Okay, I'm gonna go through this again on another slide. So if you didn't get it this time, hopefully you will the next time. MHC is always CD8 and MHC always associate with each other. So CD8 associates with MHC1. Sorry. Now, CD4 helpers, we're also going to talk about this a little bit later, but CD4 helper cells recognize only professional antigen presenting cells. And just memorize this, remember it. When we talk about professional antigen presenting cells, we're talking about macrophages and dendritic cells. Okay, there may be others. In fact, I will come back to this and talk about this, but B cells also act as professional antigen presenting cells, but we'll see how they're special in a little while. These express MHC2. Okay, so that means that if you're a macrophage, you've got MHC2 out here. Okay, you also have MHC1, which is interesting, but we don't need to talk about that. We're gonna worry about MHC2 for now. They have MHC1 because they're a nucleated cell, and if they get infected, they have to be killed too. But they have this role as a professional antigen presenting cell where they can go and present that to helper cells, T helper cells, which are CD4 cells. Okay. I feel like I spent way too long on that particular slide, but it's kind of an important concept, so I apologize if it was too long. So let's go through this again. MHC1 molecules found on every cell, every nucleated cell in the body, and they associate with CD8 cytotoxic T cells. So what happens with those? I'll just read through this. All nucleated cells express MHC1, and MHC1 presents cytosolic proteins as a test to make sure the cell is working properly. Remember earlier when I said the virus comes in and it hides inside the cell? Okay, it says, oh, nobody can see me now, I'm safe. Well, you've got this process going on in every cell of every cell in your body all the time, where it's just sort of randomly saying, yeah, okay, there's a protein. Let's chop it up into little pieces. Oh, I guess this is the protein over here. Let's chop it up into little pieces and express part of it out here. Okay, so it binds it to this MHC1, and it moves to the membrane, and it says, okay, this is what I've got inside me. What do you think? Cytotoxic T cell. Did this come from me or did it not? And most of the time the cytotoxic T cell will move right past because it's a self antigen. It's something that, it was something that was being used by the cell. But maybe, just maybe, this virus that's in there that's maybe producing a protein that shouldn't be there, maybe that ends up there. Okay? And that moves out to the outside. And then this CD8 cytotoxic T cell comes by, and maybe it's producing just the right receptor that says, whoa, you're non-self. Okay? I've been trained. I know self from non-self because I'm a T cell and I've been trained. And so I didn't mean to make that super, super long. It's okay, cytotoxic T cell. So there's the receptor, and the receptor says, I don't recognize you, kill it. Okay, so that's what it's doing all the time. Your cells are constantly sampling their own contents, putting them on MHC1, and then if a CD8 cytotoxic T cell moves through, then it will kill that particular cell. Okay. Now I remember, I didn't mention this on the previous page, but I remember CD1 goes with, or I'm sorry, CD8 goes with MHC1 because 8 times 1 equals 8. And I remember that CD4, which we're going to cover next, goes with MHC2 because 2 or 4 times 2 also equals 8. So I call that the rule of 8. Okay, so if you take your CD number and you match it up with your MHC number, it should equal 8. We only have 2 to worry about, so you could probably just memorize it, but whatever. That's the way I remembered it. Okay, so infected cells will have an abnormal profile of peptides presented, and so they will be killed. MHC1 class proteins are recognized by CD8 expressing T cells. I know repetition, but it helps, right? Or it works. You may be tired of it. CD8 cytotoxic T cells destroy the cell that activated them. Okay, so that means it's non-self. So in other words, again, if a pathogen is hiding inside a cell, this continuous sampling of the cell content will eventually reveal the infection to the cytotoxic T cells or the CD8 cells. Okay, so now we can talk about the professional antigen presenting cells. Remember, so go back to those macrophages and those dendritic cells that are recognizing these PAMPs and they're destroying or phagocytosing it. Well, they're going to phagocytose it. Okay, so if you see the difference here, now the protein or the antigen is being taken in, it's usually a whole organism at first, is being taken in, cut into little pieces, and then put out here on MHC2. Okay, because it's a professional antigen presenting cell. So this isn't interested in showing it to the CD8 cell. Okay, this isn't interested in showing it to a cytotoxic T cell. This is going to take it and show it to a helper T cell. Okay, so it's going to take it to the lymph system. So professional antigen presenting cells express MHC2. Macrophages and dendritic cells are the main ones we think about, but also B cells. We'll talk about that. Will phagocytose molecules that they view as non-self using these TLRs and that recognize the PAMPs. They then digest the pathogen and present parts of it on their MHC molecule. Okay? They then continue to the lymph system. And if there is a CD4 helper T cell that has a receptor, so we can draw one of those up here. If it has a receptor for this particular antigen, that means if it has a receptor, first of all, that means it's not self because it won't make a receptor against itself. But that means that it's non-self. And that particular T helper cell will release signals that will cause an increase in production. Specifically, what it will do is it will clone itself. Because now, what does it know? It knows that there is something out there that the macrophage found that isn't supposed to be there. Okay, some kind of foreign molecule. And it happens to have the receptor for it right there. Okay, so it goes through a lot of different receptors, rearranging those receptors, and it just found something that it stuck to. So it's going to clone itself. Okay, so in a sense, that means these antigen presenting cells are informing the adaptive immune system, in this case the helper T cell, that an infection is taking place. Okay, so T cell summary. T cells have receptors that will not bind to self antigens, only non-self. Any nucleated cell expressing a non-self antigen on its MHC1, which is all nucleated cells express MHC1 and they're constantly sampling their internal contents, will be destroyed by a CD8 cytotoxic T cell that binds to it. A professional antigen presenting cells, macrophages and dendritic cells, expressing a non-self antigen on its MHC2 will activate a CD4 helper T cell, which will then clone itself to increase in number. Okay, so what good does it do for this B cell that recognized that had a specific receptor for this pathogen? What good does it do for it to recognize it and then clone itself? Okay, so yeah, sure, you know that there's a pathogen out there and you cloned yourself. Well, I mean, it's releasing signals to turn up the rest of the immune system as well. One of those things that it turns up will be B cells. Okay. Now, B cells are also trained. They also have receptors that will only bind to non-self organisms, but they're different. If they see something non-self, they can look at an entire organism. Okay, so an organism kind of comes by. You can see that here. So here comes an organism floating through. If you have a large enough infection, it's going to make it into the lymph cells. And the B cell will engulf it. It will phagocytose it. And just like the macrophages and dendritic cells, it will rip it to pieces and then express it on an MHC2. And it will look at the T cell and it will say, hey T cell, I know I've been trained. I didn't recognize this. I just want to double check with you to make sure that this is actually foreign. This is actually non-self, because if you agree, so if the T helper cell says, yep, I just saw one of those, you know, a macrophage just brought me one of those a little while ago, look the same. It's not self. Okay, so that's foreign. Then the B cells, says, okay, I'm just checking, and then it will proliferate, it will clone itself, and it will turn into a plasma cell, and it will start pushing out antibody against that very pathogen. Okay, this is where the big immune response happens. Once the B cell starts flooding the blood, the interstitial tissue, the interstitium, or the interstitial fluid, whatever, the extracellular fluid, everywhere in the body where there's fluid, these antibodies will be. And at that point, whatever the pathogen wants doesn't stand much of a chance. So we have cytotoxic T cells that are being cloned and dividing and increasing in number that recognize it. We have antibodies that are just flooding out into all of the fluid in your body, intracellular and or extracellular and blood, and which blood is extracellular, yes, but everywhere, tissue and blood. That's going to take it down. Okay, so T cells. So unlike T cells, they find something non-self. They phagocytose it, express it. It's antigens on MHC2. And T cells that recognize the expressed B cell antigen as non-self will then activate the B cell, and by activate we mean turn it into a plasma cell. The plasma cell will secrete antibody. Okay? So in other words, B cells have to check in with helper T cells so they don't launch a full-on antibody response erroneously. It's a nice checks and balances system because it's doing something that can be potentially harmful, especially if you're a pathogen. So B cells and T cells must agree that it's non-self. Okay, so this is just kind of a summary slide. You can read through it on your own. I'm just going to read through it very, very quickly. Innate immune cells will attack foreign molecules and kill them. So that was the innate immune system. Macrophages and dendritic cells act as professional antigen presenting cells, and they will present that antigen to the I just have here CD4 T cells, which are helper T cells. So they'll present it to the helper T cells. If the helper T cells, the CD4 cells, recognize the antigen as non-self, they will clone themselves to make more. And then the B cells are also up there waiting for the pathogen to arrive in the lymph system, and then they phagocytose it, express it on their MHC2, and then present that to the helper T cells. If the helper T cell and the B cell both agree that it's non-self, then it turns the B cell into a plasma protein, and that expresses and floods the system with antibody. Now, to stop all this, first of all, in some ways it's self-limiting. These processes only go on for a certain amount of time, but there's also there are suppression or T suppressor cells that will start to be formed when the activation takes place, and then these T suppressor cells will stop that after the pathogen is gone. And then this last part, I kind of mentioned this earlier, the activated T cell and activated B cells will produce memory cells, and they hang out and they remain in the system, in the lymph node, and are readily activated if the pathogen returns. So we're talking about memory cells now. So memory cells in a secondary response. So that means that once they recognize it the first time and they say, yeah, that's not self, that's something from out there, they say, now they know that that particular organism is out there somewhere. And so they will form these memory cells, and they'll just kind of wait. So that means the primary or the first exposure may take a couple of weeks to really get through all of these things that, I've been talking forever but only an hour about. It can take the body about 2 weeks to get through that. So during that time, you have symptoms of illness, and then ultimately it figures out a way to fight it off. And then these antigen-specific B and T cells proliferate and differentiate into the effector cells. The effector cells are the activated forms of them. In the case of B cells, a plasma cell. Now, they form these memory cells, and these just kind of hang out. And then the secondary, so if this particular pathogen shows up again, it may only take 2 to 7 days for an entire full-on response to occur. It's a greater response, and of course it occurs due to these memory cells. So the primary response, which I have here, it says vaccines produce a primary response. When we inject a vaccine, okay, what we're doing is we're taking these antigens. We may take a virus that's a killed virus, part of a virus. There are different ways to make these vaccines, but somehow we're putting in someone's body an inactive form of the actual antigen, of the actual, I don't want to say the disease, but of the virus or part of the bacteria or something like that. We're giving the immune system a chance to recognize it without it being infectious. And so the immune system will respond to it, and because it's killed or it's disabled, it's not going to cause any diseases, but the immune system is activated in the same way. That's a primary response, and it will also end with the production of memory B cells, or memory B cells and memory T cells. And so next time, if the actual disease shows up, your immune system is ready for it. Okay, and no, vaccines don't cause autism. Be great if they did, because then we'd know what caused autism. All right, so antibody structure. This is an IgG presumably, but I was talking earlier about how these the receptors will change constantly. Well, there's a variable. They change constantly so they can, because you don't know what the antigen's going to look like. You don't know what the pathogen is going to be, or the antibody doesn't, or the receptor, because it starts out as a receptor. And so these are constantly changing, and then they finally maybe find a form that clicks and recognizes an antigen. So we call this upper area the variable region. Okay, that's the variable region, and it's that way because it varies. It's constantly rearranging to find to sample different pathogens. And this area down here is the constant region. Okay. Okay, so the variable region is what is continuously altered to detect pathogenic antigens. If these bind to self-antigens, they won't survive. So if you accidentally produce, I've got an antibody up here, but a B cell, but this is similar to a B cell receptor, T cell receptor. They're all very similar. If it recognizes something that's self, it won't ever make it out of the thymus or the bone marrow, depending on whether it's a T cell or B cell. Okay, so briefly going through antibodies, the different antibodies or immunoglobulins. So globulin just indicates that it's a globular protein that's associated with immune function. So antibodies are immunoglobulins. Immunoglobulin M, that's what the Ig stands for. IgM circulates in body fluids, has 5 units to pull antigens together into clumps. So it's good at reaching out and clumping a bunch of antigens together. Um, this one happens to be the antibody that forms against A and B blood types. Now we know that mom can be type O blood and have a baby that is type A blood. We know that, and that's because this larger IgM, since it's the one that's going to recognize the IgM, is not able to pass through the placenta. So it's too large to cross the placenta. So even though it can attack a blood type, it can't get to the baby, and so the baby is fine. Okay, and so what it does is that it causes this clumping and agglutination, and this is what happens when you put an antibody. You put an IgM antibody against A-type blood into A-type blood. It's going to cause all that A blood to kind of chunk together and cause clumping. So that's IgM. IgG are most of the antibodies we think of when we think of like fighting off a virus or something like that. These are what are circulating in body fluids. So most vaccines, this is the most abundant immunoglobulin or antibody. Circulates in body fluids, binding antigen, most abundant, crosses the blood vessels and the placenta, which means yay, mom can give this antibody to the fetus. She can give this to the baby, and the baby will have these when it's born. It'll have the same level of immunity to what's out there that mom has. Unfortunately, IgG is also the antigen against Rh, and this is why mom, who is Rh negative, if she starts to make antibodies against Rh positive, so if she becomes this Rh protein it can act as an antigen, and if mom's blood gets exposed, if her immune system gets exposed to this Rh antigen, she'll produce a immune response against it, which means she's going to start making IgG. That means the first baby, because the blood doesn't mix, may be okay, but if during pregnancy or at the end when the baby's expanding, the placenta is expanding, there may be some broken blood vessels in there, mom's blood does get exposed, then the next time mom has a baby, she could possibly be producing antibodies against the Rh. Okay. All right. So I think we've talked about that probably before. I know we talked about that in physiology, so you should have covered it there too, but you can review it if you want to. IgA found in secretions on mucous membranes. Okay, so that's localized there. And as we discussed in class, these are also in breast milk. That's the main antibody in breast milk. But prevents antigens from entering the body. IgD found on the surface of B cells. Okay, and acts as an antigen receptor. Okay, so this is what may be found on the B cell, but then it will when it turns into a plasma cell, we'll go through some rearrangements and turn into an IgG usually. IgE is found on mast cells. We're going to talk about how that works. But it's also, you can see here, it's a monomer, which means that it doesn't have a whole bunch of them together, it's just one. But it will sit down in, so here's a mast cell. Even though it's made by B cells, it will migrate and sit right down here inside mast cells and just wait for any pollen to drive you crazy. Okay, and we'll talk about how that happens when we talk about the immune system. So functions, some other functions, opsonization. Opsonization means that it's just kind of coating it with antibody, so we can see that here. Just coats it with antibody. And then the macrophages, the neutrophils, whatever, will recognize that and destroy it. Triggering of the allergic response, that's what I mentioned here. You can see that these IgE antibodies are stuck down in here, and then when some kind of pollen or whatever comes by, whatever the allergen happens to be comes by, it will cause degranulation and an immune response, or allergic response, I guess. Activates complement, which we're going to talk about next. It's one way to activate complement. And then clumping and inactivation of bacterial toxins. So yes, it can even recognize bacterial toxins and bind to those. Okay, so the complement system. Complement with an E, not complement. Gee, you have, great hair. So the complement system, these are a bunch of proteins, so over 30 proteins that are circulating in your blood right now, just looking for trouble. So they circulate in the blood in an inactive form. However, they are highly toxic. Specifically, they're highly toxic to mostly bacteria. Okay, so pathogens, and so they'll destroy the pathogens by creating something called a membrane attack complex. Okay, and what that does is it creates a pore. Basically, these 30 proteins that are circulating, all they need is a trigger saying right here, here's a bad guy right here, and they will poke holes in it. Okay, they go down in a very specific way. We're not going to memorize exactly how that takes place, but we have to know that the complement system does this. Okay, so it can be triggered. We just mentioned that antibodies may label the pathogen and say, hey, this one right here, complement, you need to take this one out, and that can trigger it. Or some of the complement proteins might, without even antibody being there, might recognize it as being foreign itself. Okay, so it might recognize it as being non-self and take it out that way. Okay, ultimately though, yes, it pokes holes in it, perforates the membrane, makes holes in it, and destroys the pathogen. That's what complement does. Circulating in blood, membrane attack complex, can be triggered by antibodies or just recognize the pathogen and it pokes holes in it. Okay, I think that's pretty reasonable to remember. Okay, so here's the other one that we talked about. This is part of the innate. Remember, and I'm saying this, but I'm talking about NK, or natural killer cells. Remember, it's part of the innate immune system. And I like this question because if you're a virus that's hiding inside a cell, okay, and you think, okay, I'm safe now, I'm inside the cell, and I'm going to use this cell to help me, divide. And one problem is, though, that this cell is going to keep sampling these proteins that I'm making, and it's going to express them on the outside on this MHC1. So, what if we stopped the cell from making MHC1? No MHC1, it's not going to be able to tell the cytotoxic T cells that we're here. So, the question is, what if a cell just doesn't express MHC at all? Is it safe? And the answer is no. We have natural killer cells, and natural killer cells, we go down here, we'll see, oh, you don't have MHC1. If you don't have MHC1, there must be something wrong, and it will take it out. Okay. Um, so I set that up as a virus because a lot of virus infected cells will stop producing MHC1 and the natural killer cells will take them out. But also tumor cells, when they start to become damaged, when they start to form tumors, will stop producing MHC1, and those will then be taken out. Okay, to a point. So that's another thing that cancer cells have to get through, is they have to be able to avoid the natural killer cells. Okay, so natural killer cells destroy malfunctioning cells that stop expressing self-proteins at all, so they're missing their MHC. This includes many virus-infected cells and other cells that may go on to become tumors. And then this reminder here that even though they're lymphocyte cell line, they are still innate because they are not specific. Okay. All right, so specificity, monoclonal antibodies are specific for one epitope. So remember all these epitopes? I like this picture actually because it shows an actual virus and it shows how these exposed areas are the epitopes. So the entire thing can be considered an antigen. The entire protein can be called an antigen, but these specific areas are epitopes. Now, if there's only one epitope on this one protein, then we would say that that's a monoclonal antibody. Okay, so that means, and those are good for treatments, and they're developing a lot of treatments that will form that are monoclonal antibodies against one particular enzyme that's only produced by, let's say, cancer. Okay, so a cancer cell, whatever the particular cancer cell is, may produce one protein that only it produces, and so we can take, and that would be passive immunity, we can put monoclonal antibodies that stick to one part of that one, oops, I should make that a different color maybe, one part of that one antigen, that one protein, and then can kill it, okay, without harming anything else. That's fantastic. We there are research is trying as hard as they can. This is fantastic specificity. The immune system that we have is so able to be so specific about what it attacks that we're trying to replicate that and use that for drugs and things. Now, polyclonal antibodies, which is what a lot of normal immune responses are, they may bind several epitopes. Okay, so they may bind all over this antigen, this protein. Okay. So that's what a polyclonal does. And so the use of antibodies in treatment is a growing field, and it makes sense that it would be because of that specificity. Okay, but they find out that there are problems that they weren't necessarily expecting. Okay, so last slide, fetal and neonatal immunity. Gosh, this has been a really long chapter. Fetal and neonatal immunity. Couple of things to remember here. First of all, antibody function in the fetus and a newborn is deficient. Okay, so they're capable of producing um, of capable of primarily of a primary IgM response. Okay, but they're not able to produce IgG. Now this kind of makes sense. IgM can't cross the placenta, so that means the baby, sure, yeah, let's make it able to produce IgM because that's not going to harm mom, but let's not have it attacking mom's proteins. Okay, and that's why the fetus is not able to make IgG, because the IgG can pass through the placenta. And a lot of mom's proteins, a lot of things from mom, are moving into the fetus, and you don't want that fetus to start having an immune response against it. So no immune response, no production of IgM while in gestation or before it's born. Okay. Now, does that mean that the baby doesn't have any immunity? No, we've already talked about this. The newborn has IgG levels that are near mom's level. So mom's sharing the IgG. Baby comes out immune to whatever mom's immune to. But then over about a year, the baby will start to have its own, be able to produce its own IgG. And so you shouldn't really start vaccinations. There's less point um, until after about 12 months because they're not going to be as effective because the immune system isn't quite there yet. So they're less effective and they're at they may be put at a higher risk of developing the disease. Okay, so if you have a disabled virus or something like that, well, the immune response of anyone with a good immune system should be able to respond to that, but maybe there's a chance. There's a chance, small chance, but there's a chance that the baby could develop the disease. Okay, well that's it for this section. So I hope that was kind of clear. I don't know that it was, but anyway, you can get a hold of me if you have any questions.